Ngalakin Khadijan Nyunga Boja.
Okay, hello. Today's lecture is going to focus on the CO2 fixation reactions of photosynthesis. So that second half of the whole grouping of photosynthesis. So as usual, there's some suggested readings here from the textbooks. And then on the next slide are some learning outcomes. Again, use these learning outcomes to supplement your study and quiz yourself. Turn them into questions to quiz yourself. So as I've said, the carbon assimilation reactions or the carbon fixation reactions, whichever way you like to call them, and there are some other names that we'll talk about in the next slide that also describe this set of reactions, is the second part, or you can think about it as the second part of photosynthesis. And we consider it the second part of photosynthesis because the reactions of carbon assimilation use the products of the light reactions. Okay, so you can't have carbon assimilation without having ATP and the NADPH that's made from the light reactions, and that's illustrated in the cartoon on the right-hand side of the slide. Again, the reactions take place in bacteria, the algae, and the plants, and that would be the photosynthetic bacteria, okay? And more specifically, the cyanobacteria, because there are some bacteria that do a different type of photosynthesis that don't include CO2 assimilation in the way that we're going to be talking about it. Okay, so these reactions are where carbon dioxide comes into play and the formation of carbohydrates is done. So where are these reactions taking place? Well, in the chloroplast, as you would expect, but they take place in the stroma part of the chloroplast. So remember, the stroma is the region of the interior of the chloroplast not taken up by the thylakoid membranes. And so the cartoon on the right-hand side of the slide illustrates the light reactions in the sense that light energy comes in, hits the thylakoid membranes, and you can see a stack of thylakoid membranes or granum there, and then producing the products of the light reactions, namely NADPH as the reducing power and ATP, and those two compounds are used to convert carbon dioxide into sugars or carbohydrates. So in different types of plants, it's very easy to see the chloroplasts. And so we're going to be talking about C3 plants today, and the reason they're called C3 plants will become clear in a minute. And then in the next lecture, we'll talk about C4 plants. And again, I'll tell you why they're called C4 plants in that lecture. But these two micrographs that are shown towards the left-hand side of the slide are of the leaves of a C3 and a C4 plant that have been cut transversely, so they're cross sections through the leaf, and the chloroplasts are easily identified as these yellow regions inside the leaf. So in the C3 leaf, lots of chloroplasts basically all through the interior of the leaf, whereas in the C4 plants, the chloroplasts are much more localized, and you can see them as forming rings in these structures throughout the section of the leaf. All right, so as I've said, the names of these reactions that we're going to be talking about today are quite varied, and they're listed at the bottom of the slide. So carbon fixation reactions, the carbon dioxide fixation reactions, carbon assimilation reactions. They're also known as the dark reactions, but most people are not using that phrase anymore because it's basically a misnomer. It's not really telling the truth because these reactions are taking place in the light. They're dependent on the light reactions, so they're taking place in the light. The reactions are also known as the reductive pentose phosphate cycle, so you'll see that phrase in some textbooks. Not so much anymore, but that's more of an older phrase. And then the Kelvin cycle, which is used in the cartoon on the right-hand side of the slide. That was one of the investigators who worked out the pathway. And sometimes it's called the Kelvin-Benson cycle for the other person who was involved in those early experiments working out the cycle. And then the phrase that I'm going to be using is the one that a lot of people use now working in the field of carbon fixation, and that's the photosynthetic carbon reduction cycle. Unfortunately, in a way, it's got the same abbreviation as PCR, okay, the polymerase chain reaction, but we use it nevertheless. Okay, so the photosynthetic carbon reduction cycle, or the PCR cycle, describes these carbon assimilation reactions. Okay, so what are these reactions? Well, there's actually 13 different enzymes catalyzing 15 different reactions in the chloroplast stroma. And those enzymes and the reactions are shown on the right-hand side of the slide. Now, if you can remember all of those reactions and all of the enzymes that catalyze them, then you are going to be on top of any question that I can ask on an exam. Okay? I'm joking. Okay? I do not expect anyone to remember these 13 different enzymes and the 15 different reactions they catalyze. Okay? That is not part of this unit. Right? The point of these reactions is that they synthesize the building blocks for carbohydrates, okay, for sugars, but also for amino acids and fatty acids. Okay, so more takes place in the chloroplast than just the formation, the synthesis of carbohydrates. And the reaction also regenerates the initial substrate. So it truly is a cycle, and that's why PCR cycle is a very descriptive name, the photosynthetic carbon reduction cycle. So I would like you to remember this information, okay, in text on this slide, not the individual enzymes or the reactions, but I would like you to remember the next slide and the reactions that are highlighted, and we'll go through those. So on this slide, we can see that the PCR cycle is broken down into manageable phases. So there's the carboxylation phase at the top, so we'll start there, and then the reduction phase, and then there's the regeneration phase where that initial starting material, okay, one of the initial substrates has been regenerated, making up the cycle or completing the cycle. All right? So, in the next couple of slides, I go into detail about the steps and the enzymes I'd like you to remember about the PCR cycle. So, the first phase, the carboxylation phase, consists of one reaction, one enzyme. And that reaction is the carboxylation of ribulose 1,5-bisphosphate, or RUBP. Okay? And that reaction is catalyzed by ribulose 1,5-bisphosphate carboxylase oxygenase, or Rubisco. It is fine with me if you remember RUBP and Rubisco. Okay? It's okay if you don't remember the full names. All right, they're a mouthful, which is why we've come up with the abbreviations RUBP and Rubisco. Much easier to remember, much easier to say. So Rubisco, as shown in the diagram, attaches CO2, atmospheric CO2, to RUBP, ribulose 1,5-bisphosphate. The resulting product is a very short-lived 6-carbon compound that is immediately processed into 2 3-carbon compounds. So the first stable product of the PCR cycle is a 3-carbon compound, which is why the plants using this carbon assimilation pathway are known as C3 plants. Okay? And we can see that this 3-carbon compound, 3-phosphoglycerate, 3-PGA, okay, is the first compound, stable compound, that's made by the cycle when we basically feed plants radioactive CO2. So we give them, and that's shown in the graph on the bottom right-hand side of the slide. So we feed plants 14 carbon CO2 and then chase that 14 CO2 out of the plant with air. So that pulse of 14 CO2 is only for about 30 seconds, okay, or shorter. And then the first peak that we see is 3-PGA. That's the first stable compound, and that's a 3-carbon compound, as I've said. Later on, in this chase period, when air is given back to the plant, we can see the radioactivity coming up in sucrose and also in starch, which of course is polymers of glucose molecules. So the 6-carbon compounds, the carbohydrates that are eventually made. But the first stable compound is 3-PGA. Okay, so that's it for the carboxylation phase. Single reaction catalyzed by Rubisco. The next phase is the reduction phase, and there's only 2 reactions in this reduction phase that I'd like you to remember. And this is where 3-PGA is phosphorylated to 1,3-di-PGA. Okay, so 1,3-diphosphoglycerate. The reaction is catalyzed by a kinase, as you would expect, and so that's phosphoglycerate kinase to be specific, and that enzyme uses ATP that comes from the light reactions. So here we've got the first case of where the products of the light reaction are used in the CO2 assimilation reactions in this first step of the reduction phase. And then from there, the 1,3-diphosphoglycerate is reduced to glyceraldehyde 3-phosphate, and the enzyme that catalyzes that reaction is glyceraldehyde 3-phosphate dehydrogenase. Okay? And this is where we see a second product of the light reactions being used, and that's the reducing power in the form of NADPH that's made in the light reactions. Okay? So that's the reduction phase, using the products of the light reaction to catalyze the 2 steps, first a phosphorylation step and then a reduction reaction. The final phase of the PCR cycle is the regeneration phase. Now this is a really complex part of the cycle. 10 different enzymes are involved, and they catalyze reactions that are not shown in the simplified diagram of the PCR cycle. Okay? For this regeneration phase, I'd like you to remember just the final reaction, because that's what regenerates the RUBP, okay, one of the initial substrates, in addition to the atmospheric CO2, the initial substrate of the reaction. And again, ATP from the light reactions is used in this last reaction of the PCR cycle. So here we see ribulose 5-phosphate being phosphorylated using the ATP from the light reactions, and this reaction is carried out by phosphoribulose kinase to generate RUBP, ribulose 1,5-bisphosphate. Right? So 3 phases of the PCR cycle. So remember the names of those phrases, and remember the names of those, 4 reactions that I'd like you to remember in those different phases. And they're the major ones. And I say that because that's where we see the products of the light reactions being used. Okay? Linking the light reactions in the CO2 fixation reactions of the pathway. So putting this pathway together and thinking about it in context as far as using atmospheric CO2 to make carbohydrates, we have to think about how many CO2s are put into the reaction and then what come out of those reactions. So when we see 3 CO2s, that's really bad English, when we see 3 CO2 molecules being joined to 3 RUBP molecules, in other words, when 3 CO2 molecules are fixed to 3 RUBP molecules, then we see that 5 out of every 6 of the glyceraldehyde 3-phosphates that are produced, okay, the 3 PGA molecules that are produced, along with the ATP, 5 out of every 6, okay, is used along with ATP to regenerate RUBP. Okay, so most of the products of a single turn of the cycle are used to regenerate the substrate RUBP. Only 1 out of every 6 of the 3 PGAs that are made are used for the synthesis of carbohydrates, amino acids, and fatty acids. Okay? And again, that's illustrated in the diagram. So we can see the stoichiometry in the diagram, 3 CO2s going in. We can then follow those carbons through the carbon assimilation reactions, and we see that 3 carbons come out. Okay? 1 molecule of 3 PGA is taken out of the cycle for those other biosynthetic processes. 5 out of every 6 of the carbons in that entered go back to make the substrate RUBP. So that means then for a plant to synthesize 1 hexose molecule, so 1 6-carbon sugar, it requires 6 CO2s, 18 molecules of ATP, and 12 molecules of NADPH. So it's relatively energy expensive to make a single sugar molecule. So how is this cycle regulated? Well, as you might expect, it's regulated by light. Okay? It's active when light is on, when it's daylight, okay, when plants are collecting sunlight. And we can dissect what's happening when the light is on, okay, when it's daylight. Think about what's happening in the chloroplasts. Okay, think about the light reactions first. Okay, so go back to the light reaction. Think about what's happening there when light hits the chloroplasts and the thylakoid membranes. Remember, we see an increase in the pH of the stroma, and that's because of the electron transport chain and the pumping of protons across the thylakoid membrane. Okay? So the pH of the lumen decreases, the pH of the stroma increases. Okay, remember that? Now, to balance the protons going into the thylakoids, magnesium ions are transported out of the thylakoid lumen, so across the thylakoid membrane into the stroma. And that's to balance the charges, okay, across the thylakoid membrane. That high stromal pH, because of the proton pumping and because of the increase in magnesium in the stroma, activate Rubisco and other enzymes of the PCR cycle. And the one that's illustrated on the bottom left-hand side of the slide is one of the enzymes in the regeneration phase, and that's fructose 1,6-bisphosphatase. And I chose that enzyme because it very nicely shows activation at increasing pH, which is what happens in the stroma during the light reactions. So at pH 7, yes, we see some activity of FBPase, but then at pH 8, which is what we see the stroma pH being during the light reactions, we see quite high activity of this regenerative step in the PCR cycle catalyzed by FBPase. So I'd like to go back and talk about Rubisco for the last little bit of the lecture. Now, Rubisco is not a great carboxylase. Okay? It has a very low turnover rate for an enzyme. Only 3 CO2 molecules are fixed per second for every Rubisco molecule at 25 degrees. Okay? To compensate for this, plants make a lot of Rubisco. Okay? About 50% of the soluble protein in the chloroplasts of plants is Rubisco. Okay, that's a lot, right? In addition to that, Rubisco is the most abundant protein on Earth. there's about 40 million tons of Rubisco, okay, on Earth. That works out to be about 5 kilograms of Rubisco for every person on Earth. Okay, so just to put that in perspective, that's a lot of enzyme, right? As an enzyme, it's composed of 8 large subunits and 8 small subunits. The large subunits are illustrated in the diagrams on the right-hand side of the slide in the green color, both the light and the dark green, so 8 large subunits. And that's where the catalysis happens. That's where CO2 is fixed to RUBP. Each of those large subunits is about 56 kilodaltons. The small subunits are represented by the orange and red parts of the diagram, and the small subunits are not involved in catalysis, but they perform a structural role, keeping the holoenzyme together and allowing it to function. And each small subunit is about 14 kilodaltons in size. I don't expect you to remember the sizes. It's more relation to one another. Small subunit about 14 kilodaltons, large subunit about 4 times bigger. Okay. All right, so why do I say that Rubisco isn't a great enzyme? Yes, it's got a low turnover rate, but if you think about Rubisco as an enzyme for photosynthesis, it's got some defects, okay? Maybe that's too strong of a word. It's got some issues. And that's because it has both carboxylase and oxygenase activity. So if we go back and think about the full name of Rubisco, ribulose 1,5-bisphosphate carboxylase oxygenase. So the name already tells us it's got 2 different activities. And those 2 activities are illustrated in the diagram at the top of the slide. So we've already talked about the activity with respect to carboxylase. So that's following the lower part of the diagram. So starting on the left-hand side, there's the substrate, RUBP, ribulose 1,5-phosphate. And then the carboxylase activity, adds CO2 to RUBP. And the first stable products are 2 molecules of 3-PGA. Okay? And that's the PCR cycle. That's photosynthesis. That's what we've been talking about. But the issue here is that Rubisco also recognizes oxygen, and oxygen settles into the same active site as CO2. So there's competing reactions happening here. The same active site will accept both CO2 and oxygen. When oxygen settles in the active site of Rubisco, then we see it being fixed to RUBP, and the products are a single molecule of 3-PGA plus a molecule of phosphoglycolate. So a 2-carbon compound. And that 2-carbon compound, phosphoglycolate, sets off another set of reactions that is known as photorespiration. It's also called the PCO cycle, okay? And it's also known as the C2 cycle. So again, different names, but all describing the same set of reactions. So Rubisco has this dual activity. How is it determined whether it's got the carboxylase activity at a particular time or the oxygenase activity at a particular time? Well, that is determined by the concentration of CO2 and oxygen in the chloroplast, okay? And that is dependent on temperature. So at 25 degrees, and atmospheric concentrations of oxygen, which is about 21%, and CO2, which is now about 0.04%, okay, and as you know, increasing. Under those conditions, 1 oxygen molecule is fixed for every 2 to 3 CO2 molecules. And so that says the carboxylase activity is favored under those conditions. But when temperatures start increasing above 25 degrees, the concentration or the ratio of oxygen and CO2 in the chloroplast increases. And this is because there's different solubilities of the two gases at increasing temperatures. Oxygen is smaller, okay, it gets into the chloroplast more readily than CO2. Under these conditions, oxygenase activity is favored. Okay? In addition to the diffusivity of the gases into the chloroplast, there's also a change in the rubiscokinetics at increasing temperatures. And under those increasing temperatures, we see specificity for CO2 being decreased. And these characteristics of rubisco basically point to the enzyme having evolved in an anaerobic world where there was no worry about oxygen being around. There was plenty of CO2, the carboxylase activity was working, but as photosynthetic organisms started evolving, started multiplying, diversifying across the earth, as they fixed the CO2, of course they were releasing oxygen. And the oxygen concentration in the atmosphere started building up. Okay? That allows us to live, it allows other animals to live, but it also induced this oxygenase activity of rubisco. So plants have to deal with that now. Okay? It's definitely a competitive reaction, whether there's carboxylase activity or oxygenase activity. So a little bit more about this oxygenase activity that leads to photorespiration or the PCO cycle. It really is the other half of carbon fixation because of the properties, the inherent properties of rubisco. Something to remember, photorespiration is distinct from mitochondrial respiration. Mitochondrial respiration is also known as dark respiration because it can be carried out in the dark at nighttime. Okay? Photorespiration cannot. By definition and from its name, you can tell that it needs light. Right? Now, in the pathway, we see oxygen is consumed and CO2 is released. Again, you would have been able to predict that because that's what respiration is all about. When we respire, okay, we consume oxygen, we give off CO2. Right? Same for photorespiration, so same in plants. And when you look at the pathway, you can see where oxygen is being consumed, so certainly at the step of rubisco in the chloroplast, so that's illustrated in the very top part of the diagram on the right-hand side of the page. But oxygen is also being consumed in the peroxisomes, which are involved in photorespiration. And then CO2 is released in the mitochondria, the third organelle that's involved in photorespiration. Right? So a very complex pathway involving 3 different organelles, and there is no way that I am asking you to remember all of these steps of photorespiration. I would like you to remember that 3 different organelles are involved, that oxygen is consumed, and that CO2 is released. And to remember that the CO2 that's released was once part of, the carbon in that CO2 was once part of the ribulose 1,5 bisphosphate that was used back here in the chloroplasts. So that means that if the CO2 that's released is refixed, it requires energy. And that energy is in the form of ATP. Okay, so the regeneration, or the reuse, if you want to think about it that way, of the release of the CO2 in the mitochondria, that CO2 diffuses back into the chloroplast and then can be refixed by rubisco. But to do that, there's also a cost of ATP to regenerate the 3-phosphoglycerate, to regenerate the RUBP. Right? Interestingly, photorespiration is not found in ancient C3 plants. Okay? So again, those ancient C3 plants evolved during a time when there's very reduced levels of oxygen in the atmosphere. But now photorespiration is a very integrated part of plant metabolism. And in fact, here in the peroxisomes, at the step where oxygen is consumed, hydrogen peroxide is released, and that hydrogen peroxide plays an important role in signaling pathways, okay, for other biosynthetic pathways, other stress responsive pathways in plants. And then have a look in the mitochondria, where the CO2 is released. At the same time, ammonia is released. And ammonia is used in nitrogen assimilation pathways to make other amino acids, okay, to make proteins, to make nucleic acids. Right? And photorespiration is also really important in stress protection. Okay, so if we think about a plant photosynthesizing in the light, okay, so light is bombarding the chloroplast with light energy, and that's being taken up by the chloroplast. Electrons are flowing, protons are being pumped, so ATP can be made. Now, if it becomes dry and the light is still hitting those chloroplasts, the pores on the leaf, the stomata, that allow gas to get into the leaf and gases to leave the leaf, so CO2 in, oxygen out, if those pores close, okay, to reduce transpiration of water so that water doesn't get released from the inside of the plant, then that means there's no CO2 inside the plant. The CO2 levels start dropping, okay? But light reactions are still happening, right? That means ATP is being made, but it can't be used because there's no CO2 fixation happening. That means the ATP concentrations will start building up to a point where it stops the proton pumping. And you know that electron flow and proton pumping are coupled. So when proton pumping stops, then electron flow stops. And when electron flow stops, what happens? Reactive oxygen species form, right? And those reactive oxygen species start destroying nucleic acid, proteins, membranes, and lipids, right? So here we've got a way, well, we don't have a way, plants have a way of burning off the ATP, ensuring that ATP gets used, even though carbohydrates aren't being made, okay, directly in the PCR cycle, the ATP is being used in the PCO cycle. And that allows proton pumping to continue, that allows electron flow to continue, and that mitigates the chances of reactive oxygen species being formed. So an important pathway, and like I said, the other half of CO2 fixation. So the efficiency of photosynthesis definitely decreases when plants are photorespiring, okay, when the photorespiration pathway is working. There's no doubt that photosynthesis cannot be working because they are competitive reactions, so the efficiency of photosynthesis is decreased. As I've already mentioned, the activities of photosynthesis in the photorespiratory pathway are balanced, they're linked, okay, and they are determined by the concentrations of CO2 and oxygen in the chloroplast, which is dependent on temperature. And we can see that the negative effects of temperature are mitigated by increased atmospheric CO2. So there's still that relationship between CO2 getting into the chloroplast and temperature. And that's illustrated in the figure on the right-hand side of the slide. So on the y-axis, that's photosynthesis. Assimilation, that's the uppercase A, all right? So assimilation or atmospheric CO2 fixation versus leaf temperature on the x-axis, and that's been plotted for 3 different concentrations of atmospheric CO2. And you can see that at higher temperatures, we see increased CO2 fixation, assimilation, okay, at the higher concentrations of atmospheric CO2. So high atmospheric CO2 concentrations mitigate the effects of photorespiration on photosynthesis. Plants have developed strategies to compensate for rubisco's oxygenase activity, okay, for its shortcomings as a carboxylase. and that they make a lot of rubisco, as I've already mentioned, and this is true for C3 plants. But other plants, okay, have used alternative strategies. They've evolved CO2 concentrating mechanisms, and these are found in some algae, but also in some land plants, the C4 plants and the CAM plants that I'll talk about in the next lecture. And these CO2 concentrating mechanisms, or CO2 or CCMs, they're also known as CCMs, CO2 concentrating mechanisms, are ways to reduce the effect of the oxygenase activity of rubisco on a plant and basically decrease photorespiration in the plants that have these CO2 concentrating mechanisms. And that's all for this lecture.